amphipod. Round
This one doesn't
2.
look like it's getting free.
Step away for a minute.
All this action.
Chaos.
Yeah, so we're still trying to, so Scott, France, Tammy Frank, and Mike Vecchione are on the chat room trying to figure out what kind of amphipod this is. And I think it is, Mike agrees and Tammy agrees, we think it's a Themisto type Hyperiid. But Scott, I think you're still correct in that that Themisto is maybe more benthopelagic. I've seen papers where they have swarmed near the seafloor, even though I guess generally Hyperiids and maybe generally even that members of that genus are water column based, but sometimes they swarm near the seafloor.
Okay. 30 meters.
But I remember when we were doing some midwater observations using the HOV Alvin, we were attracting with the lights of the HOV
you're at 205.
huge swarms of curls and these Themisto-like Hyperiid amphipod.
205,
so they definitely do get attracted to the lights of the vehicle.
Bridge, RV Nav.
Go ahead, RV Nav.
Can we get a ship move, please?
Sure.
30 meters at 2050.19.
And Jill Bjork said that they have seen swarms of these near the Baltimore seeps and ended up collecting a bunch of them in sediment cores.
Oh, wow.
So I guess they must be rather benthic.
Yeah.
At least at times.
RV Nav bridge move initiated.
Copy.
Okay, pushing forward. Get out of this.
Yeah, good point, Scott, about on the tube anemone, it might have spirocysts, which are sticky cells more commonly or frequently than nematocysts, which are the stinging cells. And so maybe that is how the tube anemone relies on capturing prey as stickiness rather than poison or
stinging.
It's a big crab on the left.
Looking for one of those whitish objects from earlier.
Is he talking about the thing with the siphon? But
yes,
with the
that guy's gone.
Fish just made himself
interesting.
a hole.
He's there on the lasers.
I see something burrowing in there.
It's one of those black belly.
Black belly? Black belly rosefish?
Yeah, it looked like a
okay.
black belly rosefish.
Maybe it had a pot belly too.
Look like you've got the crab at the end of the trail. Different crab or spot lobster, maybe.
There's something long and thin in the water there too.
There's
Do you
that
see that?
crab on the
White?
just off the screen
It's
to
not
the
white,
cancer
so I
crab.
don't think it's lined. It was
And
right in the
another
middle of the screen.
Munida
I
valida,
can see it.
I was
a
trying to
couple
keep
of
an eye
black
on
belly
it.
rosefish.
You'd have to burrow a little just to have a place to sit for a few minutes.
What's this, Roland?
1 minute. 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 1 min
was striped fish from before.
Yeah, okay. Yep.
And the eel pout are back.
Seem much straighter than we've been seeing them. And back to starboard.
Yeah, see the trace there? That's where the cancer crab just went down slope.
Mm-hmm.
See his track marks.
You can see another anemone top edge of the screen.
Yep.
They're all over this slope.
And going back to the Serianthid and how it goes about getting its food, Tammy Frank noted that they don't need to penetrate the exoskeleton because the gills are freely available for the stinging cells of the Serianthid to hit. And here we have another crab and anemone association. I would imagine that the crab has got a good situation there because he has the scoured out area that the anemone's tube probably creates, but then he also, as we just saw, kind of gets the leftovers of anything that doesn't get eaten by the Serianthid, by the tube worm. The tube anemone will just kind of fall down right to him.
That we passed earlier. Come down. Yeah, it looks like sediment scour. You can snap. Yep. Alright. Moving on.
It's interesting. Just looked like a little bit of the darker, deeper, reduced sediment, but somehow got to the surface without any noticeable burrowing or...
Another Munida valida squat lobster. Bob Carney was pointing out differences between the Gulf of Mexico in here, and the seep communities that we're seeing are shallower than those in the Gulf of Mexico. Of course, we haven't seen a seep community yet. All we've seen is a bubble plume or seeping activity. But the bacterial mats don't have the depth limit in the Gulf of Mexico or elsewhere compared to the other community species.
Tonguefish, yep.
Oh, tonguefish, that's right. Go ahead.
Possibly.
Oh, wow. I think this is our first tonguefish we've seen.
It is flat, okay.
Stand by one.
Wow, it's a great shot.
It's doing the wave.
Let's get it with lasers.
Lasers.
Just about a little over 10 centimeters.
Leo's clear.
Maybe 12. Oh, look at the track that he's made.
Look at that with the little ridges on the edge.
Big shrimp sitting on the sediment.
Definitely fine-grained sediments to be able to see that kind of mark.
Another anemone on the left. And some deeper holes here.
This one has something rolling.
Black belly rosefish.
Yep. All right, come on.
One example where the common name is probably actually longer than the Latin name.
Yeah.
What is the Latin name? I don't remember.
I don't know. I'm just guessing.
Helicalenius
Oh, you do know.
dactylopterus or something like that.
Okay, maybe I was wrong. That's pretty long.
You were
Ridiculous.
just saying it was a long common name.
Yeah.
Yep, Helicalenius dactylopterus. Just rolls off the tongue, like all of these names.
Can we do a quick zoom of that crab?
Going off screen,
oh,
right?
It's not a crab. That's why I didn't understand what it looked like.
Shrimp or crab.
Still on it?
There's a crab in the
Yeah.
upper right.
Let's snap video.
No shrimp. Yeah.
It's like the one we saw earlier, I think.
Yeah.
Let me know when you're clear, video.
Video's clear.
Just look at the micro scale of the sediment grains just rolling. These rosefish are the same size as the eel pouts here. Usually they're much bigger.
It's another sea star, maybe.
Mm-hmm.
I feel like our sediment just picked up.
Wow, super steep.
Second tonguefish on the left.
What's this? Is this another crab?
Yep, crab.
Looks like it.
sure.
Let's snap real quick. Confirm. Yep.
Yeah, partially buried.
All right.
No? I thought it was too, but he's just short. Oh, his
Backside is
backside
buried.
is buried.
And there's another tonguefish in the lower left. Nice track.
Crab.
Well, it certainly looks as though the
Really wanna
black belly
find one of
rosefish
those
is making many of these holes.
mollusk, maybe, things that we saw earlier.
Yeah, I think you're
Can we get a quick zoom
too
on that?
far up
Oh, is
for
that just a
Yeah.
rosefish?
starfish.
I can't quite tell.
Or is it a sea star?
Go ahead.
Yeah, it's one of those burying
Yeah.
sea stars.
A mud star. See if we can remember.
Was
Yeah.
it Gonios?
Oh yeah, we're stopped.
Gonios, Jared? Or that was a different group? I don't remember.
Let's see. The mud stars.
Yeah, do you want a 0.2 or 0.1?
Hexolicidae is the order.
Is that good enough for
Plutanaster,
you video?
perhaps?
Do you
They're the
wanna
ones
set
with
down?
the
I think that's close
pointy
enough.
2 feet.
Yes.
Tapered.
Yes.
I
Yeah.
can't see any more
But I don't
details
know
on
if
that.
that's
Yeah.
Ready to drive you nav?
the same
I
species.
figured we didn't need to sit
Maybe
down.
Chris Ma will hear our
Go
call.
ahead, RVNAV.
Hey, we'd like to put another ship move in.
Sure.
It's going to be 30 meters at 205, 0.19.
30 meters at 205 speed decimal 1.
Sorry, make that decimal 2.
This is it, right,
Decimal
Roland?
2. Okay, we'll do.
No, it's a crab. Shell.
Oh.
Molt.
Think that little crab came
Oh.
from that big one?
Aren't
the-
they supposed
yeah.
to get
Power
bigger
up
when they
suit.
come out?
RVNAV bridge, moving to
It
ship.
molted and got smaller?
Come on.
It's not right.
This is a 1 step forward and 2 steps back.
A bit of a ledge there in the
environment.
upper right.
Climbing this steep slope.
It's a lot of little black belly rosefish here.
They really are tiny too.
Yeah.
What happened to that hard target that used to be on my sonar?
The eel on the upper right we haven't seen.
What's that up in the middle of the screen?
There's one way off port too. Maybe we came up over it.
This
Go
is
ahead,
just
video.
another of the same.
It's an appenbranchid
Yeah,
eel. What
yeah.
I don't know what the one to the left was. Was it just partially buried?
That's what I was wondering. It was
Quite a
shorter.
large-
but-
Or wait, is that?
Things in his
It-
front.
That might. That looks
it's a
hagfish. I was gonna say that looks
hagfish.
Lovely.
disgusting, like a hagfish.
Yes.
Oh, we were
There you
just,
go, Scott France, your favorite.
Yeah.
These are not as pretty as the hagfish that we saw in the Lophelia mounds. I was wondering, that was a very unusual behavior for a synapobranchid eel.
Yeah.
Probably no calming him down, is there?
Hagfish are the most slimy creatures on the planet.
A pretty crab.
Bathanectes
digging in.
crab.
Oh,
Wow,
yeah,
look at
nice.
that.
Maybe he found something
Slime his
under
way
the
into the sediment.
to eat.
I wonder what's in there that he's finding to eat.
I think probably just getting away from the light.
He really is getting in there, isn't he?
Mm-hmm.
That must be what the other one off to the left was too. It was
Yeah.
like half buried, just
He's
the
going all the way in.
distilled portion hanging out.
Wow.
So that's crazy.
That's pretty cool.
He thinks we can't see him anymore.
If we
He
can wait
stopped.
till he disappears.
I don't think so. I think this is probably. I don't know. What I think of when I think of hagfish is like ripping off, like scavengers and ripping off big chunks of flesh and tying themselves in knots on
Right.
like
Yeah.
big food falls. But
Sure.
that's probably just what sticks in my head, and I don't know what their normal lives are like.
And he's definitely way in the sediments.
The hagfish burrow. Who would have thought? It's amazing. I wonder.
The ostrich hagfish. That's funny, Mike Vecchione.
Still going.
I wonder if their burrows stay as burrows when they exit. If they like are, if they produce a bunch of mucus as they go in there, it might have the sediment stick together and be the start of a larger burrow.
Could.
Or maybe not. I don't know.
I think he
Just
has
wondering.
stopped.
I wonder where he will come out. I wonder if they just back up.
And look at the other one.
The other one's gone.
Okay, so Bob Carney said that as far as he knows, they spend most of their time doing this until some kind of stimulus comes around, and they'll
Doesn't
go
seem
check
like
that
a
out.
safe thing to
Okay.
be sticking
I guess
Now
it
we
is
know
a
if we-
behavior, but it was unknown to me.
If we see that sticking out of the sediments, now we'll know that it's the tail of a hagfish. That would stump us, I think, otherwise.
75. Snap here.
Like that,
Yep.
on cue.
So Bob, are they? If that's sort of the
Sure.
normal
Just leave it.
behavior, are they feeding there? Or are they just waiting?
They're going to be the
And
snipe
there's
eel
another
image
snipe eel.
today, since we haven't gotten one.
ROV nav bridge
Go
movement.
ahead.
Try to get his
Did you ever
head.
go on snipe hunts when you were at summer camp? And
Yep.
it was always a fictitious animal. So, they do exist. That's where the hagfish is right there.
Now looking at you about 890 power.
That's all I can give you video.
Those are some long, skinny
Yeah,
bodies.
I like them.
I do not.
You don't like the snipe eels?
No.
They just come back on.
I have a snake phobia.
But you don't mind the hagfish?
2-ton fish passed.
I didn't say.
Oh,
I
you don't
didn't
like
mind
any
them.
of
I
them.
don't like the hagfish at all.
Okay.
If requested to collect one, I would say no.
Yeah, I guess the manipulator could pull a hagfish out of its burrow like that. Hold on
You
to
think
its
it
tail
would
and
slide
pull.
right out,
I
though?
can't
Yeah,
imagine
just
being
like
able to hold
probably.
on.
Mucus. Slip out.
We might
Probably.
chop it.
Probably
Yeah.
wouldn't be able to keep it in the box.
Oh,
Yeah, that's
please,
true.
let's not talk about it.
You know, that's just going to encourage us.
Yeah,
We're
I
having
know.
hagfish for lunch. You didn't know?
Disturb the scientist day.
Let's retrieve it from the box.
Something
looks like-
hard on the right. Look at the hard structure.
Something behind the Serianthid, or
Well, there's a crab and a...
but yeah, there's
Oh, more directly behind it, you mean?
yeah, kind of buildup of sediment,
It's
or
his
Go ahead.
tube.
Is that the
It
tube
does look
like
like
what we
it.
saw before?
oh, maybe it is just the tube.
Where the tube is fallen down a little bit.
Well, that
That tube looks a lot thicker than it should be given the size of the anemone, though.
it's big.
Unless it's just covered in... I guess it could be covered in lots of layers of sediment.
Yeah.
And that's a very
Here's
tiny
a-
Bathanectes crab, too.
That was
Here's a question for science. So,
It's
since
wide.
these anemones are tube anemones, and we see the tube, sort of gives the thought that maybe these animals are larger than I've always thought. Do we know how long the actual living tissue?
Yeah, they're fairly long, and especially the tube itself. So the tube could be like even a meter long, way back into the sediment, and the animal would be a portion of that. I would imagine probably a foot and a half or so is probably how long the actual tube anemone
Another
is.
tube on the left.
And I've only seen one of them, so I don't have a good sample size on that. But
Can we get a zoom in of the sea star?
You got it.
Be good to get an ID on this. Get a good shot of him without the sediment on top.
so Jill Bork was talking a bit about Baltimore seep, which is north of us and at a very similar depth. And mussel beds and bacterial mats were found there with higher abundances of infauna associated with those compared to deeper seeps. So if we do see a seep community here, there might be quite a bit of diversity
Okay, go ahead.
in the sediments. That's what the infauna refers to. And Jill and Amanda Demopoulos work on the infauna. They've been in the Gulf of Mexico, all over the place, as well
Look at his
as
little walking
along
feet.
our
Look
coast
like
here.
They came from below. See his tracks?
Thanks, pilot. You can see its sieve plate with a madreporite.
You can see the sediment spilling down,
Yeah.
too.
Those tube feet are rustling up the sediments underneath.
Be interesting to see if this guy has the pointy tube feet that the mud star that we saw a couple days ago had.
Maybe he's burying himself now.
Might be, yeah. Yeah, you see the sediment starting to coat him.
Video's clear.
Coming up.
And Jill adds that the bacterial mats are generally characterized by high abundance but low diversity of the taxa that can tolerate the sulfide concentrations.
Keep it moving.
Great. Now, if you could do it at 0.1 too, 0.1.
Mussel sediments actually had high diversity.
Let's keep it at 0.2. Let's keep it at 0.1.
Okay.
Because we got lots of time.
Okay.
All right, I think 0.1 gets us to waypoint 5 in plenty of time.
Ready, RV nav.
Go ahead, RV nav.
Hey, we're ready for a ship move.
Okay, I'm ready as well.
30 meters at
Big
205, 0.19.
Okay, 30 meters at 205, speed decimal
That's
1.
a really big depression here. One tiny black-belly rosefish.
ROV nav bridge, move initiated.
It is
Copy.
deep.
And look at all the little marks around here.
Yeah. The little
Zoom to the bottom.
holes.
Little holes.
That might be from
Micro-holes.
gas.
Nope.
Okay.
Doesn't seem to be active.
Just come wide.
You are wide.
Yeah.
It's another couple larger holes. Another tonguefish right in the center of the screen.
Pushing
Want
past
to do
the
a zoom on
streets.
that?
Pilot, can we zoom on the tonguefish?
It's just to the right.
Go ahead, video.
Oh wow, that's a very pretty pattern.
There's a little
Look at his eyes at the
amphipod
bottom.
or something crawling up, just
Stand
upslope
by.
from it as well.
Wow, that's
Oh,
a
look
really
at that.
beautiful one.
Let's bring it partial. So I can stabilize.
So does their eye migrate over to this side as they mature the way that flounders' eyes do?
I believe so.
All right, let's go back in now. Ready? I thought I had it. Sorry, video.
It's great.
Okay, video's clear.
Okay.
Bob Carney pointed out that the holes all have like shaggy stuff hanging down.
And there's a little tiny, just above that, right to the left of your
Go ahead, back in.
zoom in right now, there's a little organism making a trail. Can't tell if it's a hermit crab or it's so small.
I think that is just a growth.
Did a
I think
tiny
the trail
bivalve?
continues past
I
it.
don't know. It's made a trail.
I think
going
that's
this way.
just something else growing there. The trail goes past it.
Maybe
I think
so.
that's just
Okay.
floating
Oh yeah.
Mm-hmm.
next to it.
Okay. Oh, this is definitely more pockmarked than what we've seen.
Mm-hmm.
Does that mean animals, or does that mean bubbles coming out?
I don't know.
We haven't seen bubbles, but right here anyway.
Right.
We'll keep our eyes peeled.
We have seen surfaces like this in the Gulf of Mexico where we see a sort of distribution of lots of holes, but I don't think there's any real way to delineate at this point with no evidence whether we're looking at furrows or
Fish on the
bubble
left, off
traces,
screen.
the trace of bubbles having been expelled from the seafloor.
Yeah. Thanks,
Yeah.
Carolyn.
So what's this line that I'm coming up to? Ahead. It's like a V.
Yeah, it looks like a V of reduced sediment up here. And that's interesting. It's close to all these bubble or little holes in the sediment.
So out of curiosity, I don't know if the pilot can tell us how strong the current is here.
Pardon me, Carolyn, I didn't quite hear you.
I'm sorry. Does the pilot, does someone know how strong the current is?
Oh, okay.
At the bottom.
Let me ask, pilot. Did you hear that? Do you know how strong the current is?
Watch lead. It's a bit tough to tell. As you've noticed, we're seeing a lot of topography down here. So the current is seemingly picking up but decreasing. I'm off stick right now, and you can see I'm not moving very quickly. But there's definitely been portions, like when we first got on the ridge line, that it was much higher. So I think we're I think it's all over the place.
Earlier it was flying like when we were coming up the ridge.
So it looked like the sediment came from those two holes on the right.
Okay, the pilot said that the current has been pretty variable, and right now it's not much at all. But when we got to the top of some of these features, there was more current, and they were being pushed a bit.
Okay, thanks.
Another tonguefish. It looks like a similar one with that band across its tail, back end. Is that a different fish on the lower right?
Lower right, sorry. Not the one going by.
Right. Oop, I think it's just below us now. Yeah, there.
Oh, yeah.
I wonder if that's another one
On
of
the lasers?
Or is it a
hake that buried, like we saw yesterday?
I'm sorry, watch lead. I'm not seeing where you're looking.
Okay, it's
bottom left.
Bottom
Yep,
left,
yep,
yeah,
yep.
way in the corner.
Got it.
or is it a crab?
Oh, it's a crab, probably.
Yeah,
Just,
please.
It does look like an
Partially
eye.
buried fish.
Oh no, it is
It
a
is a
berry
fish. Good,
It is that
good.
southern hake. Wow. All tucked in, ready to snooze.
Is this southern hake, is that genus Merluccius? Or is it a different, like I don't know what,
Let me look.
I don't know what hake encompasses. I know the one we have off Calavarnia is Merluccius, but I don't know. Or Maluch, I don't know how to pronounce it actually.
Touchdown.
Video's clear.
Pilot's clear.
He has a little blank.
Yeah.
So cute.
I didn't know they would bury themselves like that.
Yeah, we saw that same behavior yesterday.
Oh, I missed that.
Europhyces.
Oh, okay.
Europhyces regia.
It's the same one as we saw yesterday.
I would think so. It looked like. Well, I mean, I guess we can't totally tell
see
because
the
we can
whole
only see
thing.
the head.
Yeah, that's true.
Did chip stop?
It'll be. It's not yet. We have a little bit more, but I mean,
Video, let's snap here on this.
We have maybe 3 more minutes or so.
Yeah, I'm not sure if it's if it
Oh,
hides
it's
like
30
that
days.
to be
Let's snap on the lasers
camouflaged, so
video.
that if something comes by unsuspecting, they can
Wait till we
leap
answer.
out,
Okay,
and
awesome.
that looks like a
Dead
snake.
eel pal.
It does.
Oh,
Oh, could that be one of those nut
looks like
worms?
a Echiuran worm that is no longer with us.
Yeah, there's the
Are you
tongue
sure?
at the end.
Yeah, an acorn worm.
Could it be a live acorn worm of a different color?
Usually you wouldn't see the entire animal out of the sediment, I think.
Yeah.
And the middle section being that colored doesn't look good to me, but I don't know. That's just my guess. Oh, yeah. Enteropnust, not Ekiurin.
That's not me. The dust.
And are those?
Video swapping out.
Those are usually in burrows, though, right?
Yeah, that's what I mean. If it's not at all in a burrow, then it's probably
I thought it was.
not alive.
Well, I'm surprised it hasn't been eaten.
This one to the left, under the
looks pretty
lasers, though,
fresh.
is an eel pout, I believe. Video, let's just confirm that this is an eel pout.
ROV nav bridge, move complete.
Copy that.
Yep, yes it is.
Yep.
All right, let's come on.
Let's go
So
ahead
many eel pouts
and
today.
put another move, same move. 30 meters, 205, 0.19.
All right, 30 meters, 205.1.
It's interesting when the skids of the ROV push into the sediment, you see a little micro avalanche.
There we go. What's
and
this over here?
segments of the sediment break off in blocks, and that's very similar to what we see on a very large scale on this steeply sloped continental margin off of North Carolina and all the way up through to Nova Scotia, where large portions of the margin detach and slump down to greater depths. And actually, we're going to be looking at the toe of one of those very large blocks on our last dive. Jason Chater from the U.S. Geological Survey has been studying similar submarine landslide features, so he's going to be helping us with that dive, I hope.
Sorry, what?
This is an interesting feature. I would say it's a likely candidate.
Whoa.
Look at all that brown or gray sediment.
Lasers clear.
It's blowing away.
Thank you. Huh. And what are those little fluffy things hanging down there?
Little tubules.
Bob Carney said this is a bubble chimney.
Yeah, it definitely looks like one.
Really? Oh.
Wonder how often it bubbles.
I think we can come wide.
Wow, that's impressive.
If you've just joined us, we actually did see some bubbles erupt, just a few, but out of one of these types of pits. So that we know we are in a gas seep area, and we're hoping to find some more evidence of that.
You can take the skids out.
Some plumes small plumes that are sourced in this area. We detected several plumes on our multi-beam system as we were coming across this site this morning. You can see them in the water column, the bubble plumes, the water column of the multi-beam sonar trace.
Is that a good enough look, watch leads?
Yeah, that's great.
All right. Carrying on.
Do you like flying with lasers?
And as we...
Yeah, I was just trying to get a moment of that without the lasers.
As we do carry on, we want to give a shout out to the Broward Shell Club and Conchologists of America. Dr. Harry Lee from the University of Florida gave us an identification on the snails that we saw earlier.
Oh, great.
Family Turidae, and they might be Gemula parascleta. And that was Carol Marshall who gave us a call.
We're looking a little hot here, video.
You're talking about the 3 in a row that we saw. Okay.
Yeah.
And that's what we call citizen science. Thank you very much.
That's wonderful.
Crowdsourcing.
Mm-hmm. I wonder if the Conchologists of America saw the slit shells that we observed yesterday.
I hope you did.
Those were some beautiful shells.
If not, I'm sure we'll have pictures of them at some point.
Yes.
To show.
8.2.
This is a different fish than we've seen before today.
Let's get a snap here for ID.
It looks like the codling, but slightly different.
Yeah.
Maybe just older, bigger, darker.
Sure.
Yeah.
Any like dorsal fin? Do the others have that? I haven't seen that.
Yeah, I'm not
Yeah,
sure.
I think they
Maybe
do.
they're just
I think
folded
it's
down.
I think it's translucent though, so you don't see it as obviously.
Pretty.
Could be wrong though.
A prominent lateral line.
I'm not sure that we have very many fishy people on the chat room.
Let me know when you're clear, video.
I keep wanting to call those codlings Liponema, which is like a pom-pom anemone from the Pacific. So I don't know why I
Very
can't remember their name.
Another chimney, maybe?
Sometimes,
So
sometimes
I
something gets
apologize
stuck
for
in your mind, right?
continuing to call it codling, but that's all I can remember.
Okay, moving on.
Ooh,
What's that?
nice ctenophore. It's a burrowid ctenophore, which is more of a active predatory ctenophore. They like eating other
Looks like you came into
ctenophores.
this too quickly.
Well, I shouldn't use the word like, but
They do.
they do. Yeah, the one, they kind of look like they have pursed lips, and then that will open and envelope another ctenophore or jelly or something else, and kind of.
Looks like we have a couple of Rhinius pings on D2.
Tonguefish.
We're not seeing as much burrowing or pock marks, whatever generates those. Here it's a much flatter landscape. But lots more of the little holes.
And we're at 385 meters now.
So I think it was like about an hour ago now, maybe more, that we were going to have the front row introduce themselves.
Oh,
but
that's
then we got
right.
distracted by
Right.
bacterial mats.
That's
We
great.
have so much action.
So maybe you could do that now.
Co-pilot, why don't you start us off?
Hi everyone, this is the front row talking. We are from the Global Foundation for Ocean Exploration. We're made up of different types of engineers and videographers. We maintain, build, and operate the ROVs and also responsible for all the video. Sitting pilot right now is
Bobby Moore.
sitting co-pilot Sean Kennison, sitting at nav,
Andy Lister.
and then
And back in the back row is Annie White on clipping.
And they do such a great job bringing this video and exploration right to your computer or living room or office.
And I'm Stephanie Bush from the Smithsonian Institution National Museum of Natural History.
And I'm Cheryl Morrison from the U.S. Geological Survey Leetown Science Center.
And I'm Leslie Sauter
Ooh,
from
that's
the
a good
College
shot.
of Charleston Geology Department.
And right just about center screen, we're seeing a jelly in the water column right above the seafloor, and it looks like Solmicis, possibly, which the common name for that is the dinner plate jelly, because they can get the size of a dinner plate.
Pretty flat too, huh?
Very flat.
I don't think he's going to get up.
and they actually can
might be able to get them
ratchet their tentacles around and lead with their tentacles. So they're active predatory jellies. This one looks like it's got some of like possibly full stomach. Although mostly it actually looks like sediment, which is not what they're feeding on. So
try and tilt up to get them in the dark.
It also looks like it's missing some of its tentacles. So generally not doing very well as far as jellies go right now.
Nice.
But obviously still pulsing the bell and moving around. So yeah, unlike most jellies that are trailing the tentacles behind them, where you see the bell pulsing, these jellies can
he's coming in by light, I think.
Ratchet the tentacles around so that they're on the opposite side of the body, and so as they move forward, they're leading with their tentacles and potentially running those tentacles into their next prey.
Okay, gotta let him go.
Okay, we'll let him go right off the edge of the screen.
That was lovely.
Time to let him go, video.
Thank you, pilot. That was nice.
Very good.
ROV nav
go
bridge
for 'em?
is complete.
That's another one of the burrowy ctenophores that we saw a few minutes ago. So you can see the 8 rows of 18 rows, and there you see actually it opening its body cavity up. That's where it would open up an envelope, potential prey.
Like D2. There we go.
Yeah.
All right, let's got to get out of this and reacquire.
Ship is stopped, pilots.
Copy.
Copy. Ship stop.
And Scott Harris, who's a geologist at the College of Charleston, contributed.
Bubbles, bubbles.
Traveling, so there's some bubbles right now, and this is just timely. He suggested that,
and a squid inking like crazy.
Look at that. And so that's probably indicating these little pocks are actually bubble releases, because it looks like he's disrupting that. This squid. But as these bubbles rise, they expand as the pressure decreases. And Scott Harris estimated that a 1 centimeter diameter at this depth of about 400 meters, I guess we're a little shallower than that, but and if there's no water column loss, which there often is, but if there were no loss, would be approximately 30 centimeters in diameter at the surface.
That's
so
it's pretty hard stop, but
I never thought about it like that.
No, yeah.
It's cool.
Yeah, but let's be fair, that's based on
Carolyn, we can't hear you. I'm sorry. Can you?
It is a methane on the way up. So the bubbles emitted usually when about 100, 160 meters water depth of normal size reach the surface with no methane. The methane is
anyone,
by other
anything?
gases from the water
Bubbles.
column. And some of the bubbles don't, they burst on the way up.
So did you say what size would they be on the surface, Carolyn? We couldn't quite hear you.
We can't hear you very well.
Go ahead, video. Let's get this guy real quick. This fish.
Today, so I think that's why I don't have any stuff. Yeah, so to be fair, that calculation is
That looks like a viperfish, Coliotis.
know that a lot of bubbles don't.
Look at that jaw. Going in the jaw real quick.
They burst on the way up. Furthermore, I want to be very clear with the listeners that we don't expect any methane at all
And he's gone.
to the surface in
Nice
most
job, video.
water columns. If the pebbles are emitted,
So I think it may have been that squid again hitting the sediment.
Normal size that we think happens.
Because I'm not seeing any bubbles anymore.
From the seafloor, if it's deeper than 100 to 150
And it's
meters,
about what we saw
it will
before.
not make it to the surface with any surviving methane. That methane
Time to push through.
is replaced by other gases that are much more abundant in the water during its ascent, leaving the methane in the water column where it is available, for example, to be changed into carbon dioxide by microbes that act on that.
Oh, that's great, Carolyn. Thank you.
Direction.
We were mapping some vigorous bubble plumes off the coast of Washington as part of the Ocean Observatory Initiative. University of Washington's ship, the Thomas G. Thompson, and we saw a clear horizon where the bubbles stopped. And so they were getting, I think part of it was density stratification in the water column causing them to pause, but certainly they were not reaching the surface. So
that's also an active research area. So one of the problems, of course, is that it's a detection limit issue as well, right? So what can our sonars detect? And they may just not be detecting the survival of micro bubbles above a certain water depth. So Scott's philosophy at Texas A&M University is probably the leader in that particular aspect that combines sort of environmental engineering, acoustics, and physics.
Great, thanks for your input, Carolyn. So, Stephanie, the fish we saw, we had a nice close-up. What was that again?
Viperfish?
Viperfish, Coliotis.
Viperfish, Coliotis.
C-H-A-U. Yeah, they're one of the species, one of the many midwater fishes that have teeth larger than their mouth can handle. So they're constantly swimming around with their mouth half gaping. But that's so that they can basically catch whatever meal they come across and hold it in their teeth, because food is hard to come by in the water column.
It's starting to get out there. Still some tether, but you're running out.
Well, now you can definitely get a sense of the feature that we are climbing. We've been as close to the crest of this ridge for most of our dive. It's just very narrow here, so you can see both slopes. And we began our dive at about 510 meters, and we are currently at 380 meters. And we've seen a few areas where gas bubbles, likely methane, are being released.
Video, let's snap down here. Left the lasers.
Here we have another tube anemone, whose tube, we've heard from at least Cheryl's experience where they tried to collect one, can go down another meter or two into the sediment.
At least a meter was the one that we collected.
All right, I think we can carry on.
And that was quite a process with the suction sampler. You keep vacuuming and vacuuming. Kept going down and down. And
did it get stuck in the suction device?
No, it finally came up.
It did come up? Wow. How'd it look when it came out into the sample bin? Was it all mangled, or was it?
It was
pretty
actually
intact?
pretty good because it sucked into the tube. I think the tube kind of protected it a bit from its crazy travel through the hose.
And the tubes are pretty tough, right? Like kind of like leathery almost, or
and then a nice shiny mucousy layer around the inside.
Nice it's all mucus. About mucus.
Yeah, I wouldn't characterize that as nice.
Yeah, those words don't go together very often, do they?
Nice mucus.
We have the ship underway, right?
Yeah, so your fan keeps blowing your papers away from you. I don't want you to lose your names.
It's only moving at 0.1 though.
That's fine. I will find something to look at.
Copy. Should catch up momentarily.
And so the slit shell that we were seeing yesterday is also called a top shell, and that's a Calliastroma, genus Calliastroma. And that message came from somewhere on shore. Thank you for whoever provided that.
That was the one that was like purple with purple or purple pink, and then maybe a yellow stripe, right? Okay, yep.
And if you looked at, if you could see down at the bottom, there'd be a slit.
Yeah.
We didn't. I don't think we could see that slit yesterday, but it's such a beautiful shell that it's very well known and conspicuous.
Similar colors to the Anthias.
Yeah,
kind of
pink and yellow. Yeah. The lasers are hitting all of the marine snow in the water column.
And resuspended sediment.
Oh, yeah.
Mix.
Yep.
Apparently we can't pull that up.
This ridge has evolved into a very sharply pointed
and see if those marks were on the camera or on the monitor.
Long periods of erosion of these flanks that
up
are
in the
fairly
top left corner,
stiffly semi-consolidated sand or muds, excuse me. And
that's what I thought I just did.
I should say they're more compacted. They're not really consolidated, and that allows them to have this very steep angle.
So
call it angle of repose. But slowly they do erode with all the burrowing or the
junk.
Might just be dirt.
Bubbling that is going on below.
Make a note. Check it.
Loosens the sediment, and then gravity allows that material to slowly make its way down to the base of the canyon, and little by little it's eroding into this crest form. That's a beautiful.
Nice siphonophore.
All right, good to know.
So yeah, we're seeing this colonial organism called a siphonophore in the water column here. So on the left, actually going off screen, are the swimming bells, which transport this animal around, and then the tail end has the gastrozoids, which contain the stomachs and the stinging cells to capture prey for the stomachs, and it also has the reproductive zooids. And that might have been Bargmania, but probably I'm wrong on that. Just throwing that out there.
Wherever that ship move ends.
Still like 225?
Let's do like 2.
And we've received an update on the tonguefish that we saw previously from Tom Monroe, a tonguefish expert. And the two individuals that we saw were Symphurus maginatus. The first one was more uniform in color, and that was the female.
Bridge, ROV nav.
And a ripe ovary could be seen. I wouldn't know where to look for that, but that's really interesting. I guess we can go back and look at the picture and see if we can figure that out. And then the second individual was a male. It was about the same size but different coloration, and no ovary.